Next speaker comes to us from the University of Cincinnati Department of Pediatrics. Dr. Abbie Leino is a member of the Division of Translational Clinical Pharmacology and an assistant professor in Cincinnati.
All right, so I get to officially be the first person to welcome you to the afternoon. Thank you all for being here this morning. And so I'm going to mix it up a little bit. We're going to talk about how do we get all these samples that everybody's been talking about over the last several days to inform all of their really cool models. And if you're more of a basic scientist doing your microfluidics, as you move this towards patients, at some point, somebody's going to need to get a sample. And so how do we do that? And I'm also really interested in from the clinical side, right? So how are we going to translate all of this really cool work into therapeutic drug management? And that's most likely going to be a highly variable or narrow therapeutic index drug. As most of you probably know, getting samples isn't easy, particularly from that patient perspective. There's traffic to contend with, long lines in clinic. Venipuncture can be very frightening, particularly for our children. They're not big fans of it. And then oftentimes we only get a single time point to inform our models, which may or may not be the most helpful or adequate information. And so microsampling can help us. It's not new. Neonatal newborn screening has been around for a long time. In fact, I'm a transplant pharmacist by initial training, and there's a paper from the year I was born using microsampling to look at cyclosporin concentration. So it's been around for a really long time. But what has changed is our advances in our analytical, our mass spec, the ability to get really accurate results for those small volumes. So things like tacrolimus, where we have very low concentrations in the body. But the key thing we're going to talk about today is the advances in our sampling technology. So we've really moved from those filter cards that aren't very quantitative, that are affected by hematocrit, to more quantitative, you know what the volume of blood you're dealing with tools. And so there are a couple of different types. So there on the, your left-hand side, you have more tools that are designed around the traditional filter paper matrix, but applying microfluidics to get a fixed volume, versus on the right side are some of the more polymer-based tools that absorb blood to a fixed volume. And those are the ones that I have found to be most useful in my research. So there's lots of benefits. We could talk for several minutes about that, but I won't bore you with that. But it can be patient-centric. It can really help us increase our data. I really loved that one of the earlier presenters said, oh, I didn't have enough data to capture absorption. And if I had $1 for every time we've talked about that, I would be a rather rich woman and not probably still in academia. So we can really get an increased number of samples. In my K-12, we're actually getting 7 samples in a single day. And so patients are open to that. And we can also do lots of analytes, so also in my K-12, which I unfortunately don't have data yet for to share with you, but we're moving into the multi-omics kind of perspective of getting that data from the microsamples as well. But I'm going to focus on that patient perspective just to kind of mix things up and be less math heavy for you. And so this is some data from a mixed cohort of children ranging from 3 to adults. And we can, so we don't have a lot of data in really little kids because you can't use your face's pain scale, right? If you're younger than 3, they don't really understand the concept of that. But when we ask patients and their families, nurses and things like that, they also report similar results. But we can see that sampling from the arm, which would be the Tasso device, is the least painful. Most patients don't find that to be painful at all. They describe it just kind of as the pressure of pushing the button on the device. Whereas that pain increases more with the venipuncture. And if you look at healthier populations other than kidney transplant who are used to getting big dialysis needles, these numbers are a little bit more dramatic than what are shown here. So that's kind of the caveat of the population that I study. Device usability, so can patients actually do it? When we use the system usability scale, which is a really quick and dirty survey that asks them, like, do they think this is easy to use? Does it have too many steps? You can see that it is right along the lines of the other home-based tools that we expect them to use. So I always like to focus on the thermometer, right? Who in here thinks a thermometer is hard to use? Probably nobody. The physicians in the room probably ask their patients to take temperatures all the time. they're able to successfully do it, and even that only has a score slightly above 80. So we're not really expecting any of these devices to have super high scores. Both the Tasso and the Mitra are right in line with kind of the other sampling techniques, the glucose monitor or an EpiPen, other things that ask them to stick themselves. When we get a healthier population, the scores bump up a little bit. So adult patients who are doing it in like an epidemiological study, they find it to be a little bit easier than those brand new transplant patients I work with who are overwhelmed with the rest of the care that's going on. And we see similar trends across my work as well as others. So will patients give us more data this way? And the answer is yes. And so these are patients who are fresh after transplant, and we haven't really talked about potential benefits of getting additional data. This is just purely based on how many times would you let me stick you. And you can see for venous puncture, most of the patients are only willing to do that peak or that trough, kind of the current standard of care. But as we move to the microsampling devices, you get a lot more patients who would be willing to provide enough data for something like a limited sampling strategy, all the way up to the patients who are like, if you tell me this is going to benefit me, I would use this device every hour for the entire day to get you the data. We also know from the literature that when you ask people to do this longitudinally, maybe adherence isn't the best, using the historical dried blood spot cards. So we've also looked at how can we incorporate mobile health technology to maybe make this better. I also know the modelers in the room are thinking, I don't know that I trust the time that a patient writes in a patient diary to use that in my model. And so we used text messaging-based communication to help with some of that documentation rather than relying on this paper that gets mailed to you from wherever. And so it was really basic. This was not super high-tech. It was just done in REDCap. It was based on the time that they took their medications on an average day. So hopefully in the future we'll be able to start with some chat bots and things like that to get this a little bit more refined. But REDCap worked really nicely. So patients who were able to reschedule the collection day had 100% of their samples, all 4 samples on multiple occasions, returned to us. When it was unidirectional, that number was a little bit lower, 83, and that was really primarily related to the fact that it was counted as missed if they didn't do it within that 48-hour window of when we had planned to have it done. Whereas in the bidirectional, they could reschedule that and it automatically reset. Documentation was also much better. 100% of the bidirectional data for documentation of sample collection time was considered usable. And you also had that time stamp from the text message receipt. So you also kind of could see, they said they did it at 58, we got the text message at 59, so probably we can trust that a little bit more than a diary that we received that could have been filled out at the end of the day. Some diaries weren't received, some had missing data, some you couldn't read what the time was, so definitely getting some electronic documentation helps there. So after collecting 4 samples, what did patients think? So pain scores remained low, even after multiple pokes throughout the day. They kind of were intermediate on whether this interfered with their daily lives, which makes sense. If I'm asking you to stop what you're doing 4 times to get a sample, even though it only takes a few minutes, it does interfere with what you're trying to do. They thought it was easy, and despite the potential to interfere with their day, the vast majority would have been willing to do more than 4 samples. So again, reinforcing that they would allow us to get additional data to do things like full AUCs or more detailed collection around things like enterohepatic recycling and things like that. They do think it's acceptable. They like the idea of home collection of drug levels. And they also think that it's feasible so that they could do it. The one there that has a little bit more of the pink, which would be disagree with this statement, use the word implementable. And I think maybe that was not the best word for the patient population because this is using a tool that was really developed more for healthcare providers. And so I think in talking with patients, they didn't really understand what that meant. So we did a lot of sitting and talking, lots of interviews with these patients about what worked, what didn't work, what doesn't work in current care, and every single one of them went on a rant about the lab. Transplant patients get tacrolimus trough concentrations, somewhere around 8 o'clock in the morning because we need that to be well timed, and that's a very busy time in the lab. That's a very busy time for them trying to get to work or get kids to school for the pediatric patients. So if they didn't have to be in the lab at that time of day, they would love that. They all thought it was easy. And the other really cool thing that came out of these interviews was they understand that people are different. They may not get it 100%, right? So some of the descriptions of ADME were not the most accurate, but they get that it's different, and they get that they want to know this information. So they want to be participants in your study. And so I think if we can make things easier for that to happen, I think that would be really beneficial for both our research and for patient care. Now, we didn't talk about the analytical sides and those sorts of things, and so I know there's probably a plasma purist in the audience who's going to tell me whole blood, it doesn't give you the data that you need. And I would argue, well, is some data better than no data? And there are ways around that as well. And so I would be happy to chat with somebody at lunch if you want to talk more about that. But let's get moving to lunchtime. And so I think patients want this. They can do it. Mobile technology can help us get to that point. And I think we can eventually achieve some really cool ability for precision dosing in the real world using it. Thank you.